Okay, Lauren, can you see if we still have people joining?
We have a few still coming through, and we'll start in a minute.
Thank you.
Right, Megan, I think we can start now. Thanks.
Good morning. Okay, I'm sure if there are still some other people that are going to be joining, that they can still do so. In the meantime, we are going to get started just for the sake of time. So thank you very much for joining us this morning. My name is Megan Kotsia, and I am the product manager for IDT at Whitehead Scientific. We will be recording this morning's session as we did with the last one. And at the end of the three-part series, we will be sharing the recordings with you if you should wish to visit them. Okay, so if you have any questions, please feel free to type them into the Q&A box. We will answer these at the end of the session, or alternatively, if we are not able to answer them within the session today, we will get back to you in person. So the topic for today is xGen, NGS Hybridization Capture, your flexible solution for targeted sequencing. And this is going to be presented by Julia Duda from IDT. So IDT, or Integrated DNA Technologies, is a supplier of custom nucleic acids serving the areas of academic research, biotechnology, clinical diagnostics, and pharmaceutical development. And our presenter today, Julia, is a member of the IDT NGS field applications team in Europe, where she provides support to researchers using IDT's range of xGen NGS solutions in the field. So I am going to hand over to Julia now for her to tell us a little bit more about hybridization capture. Thanks, Julia.
Thank you, Megan. Thank you for kind introduction. Yes, so this is my camera on, and I will share my screen. If you can just briefly confirm that you can see my screen.
Yes, thank you.
Great, thank you. So yes, welcome everybody. Without further ado, I will begin. As 2 weeks ago, I will start with brief introduction of IDT. So apologies to those who attended the last session, and for those who are joining us for the first time, I will just briefly go through some facts about our company. IDT was founded as a spin-off from the University of Iowa in 1987 and quickly became a leading brand in DNA and RNA synthesis. We are, however, constantly expanding our portfolio to cover more and more aspects of molecular biology research. Today, we are more than 1,500 associates across 11 locations. Our headquarters are based in USA, in Coralville, but we also have production facilities in Belgium and Singapore. We leverage our expertise in DNA synthesis to develop innovative products for many applications, such as functional genomics, synthetic biology, qPCR and PCR, CRISPR, and of course, next generation sequencing. Our NGS portfolio consists of products that will convert purified DNA and RNA input samples into sequencing-ready libraries, including various library preparation kits, solutions for targeted enrichment, and library normalization. Many of our protocols were also automated on several automation platforms. In this busy slide, we can see a summary of our NGS portfolio, and main products are listed at the bottom. 2 weeks ago, I was discussing various library preparation methods. Today, I would like to focus on hybridization capture as a solution for targeted sequencing. Hybridization capture capacity for mutation discovery makes it particularly suited to cancer research. And at the webinar, we will discuss IDT xGen hybridization capture offering, which includes a variety of predesigned panels, custom panels, and they are available in a range of panel sizes. So firstly, let's start with explanation what targeted sequencing is. Targeted sequencing methods allow for interrogation of specific regions of the genome so that researchers can obtain information about the genetic material they care about the most. Targeted NGS workflows, the most commonly used are amplicon sequencing and hybridization capture, and they generally require less sample input and they result in data on specific areas of the genome, meaning researchers get more manageable data sets for in-depth analysis. Now, 2 different paths can be followed if you want to enrich for regions of interest. In this section, we will discuss target enrichment via hybridization capture. And in this method, DNA and our RNA samples are converted into whole genome or whole transcriptome libraries, and then they are followed by capture. This yields an enriched library that can be then sequenced on various NGS platforms such as Illumina, IonTorrent, PacBio, and so on. Amplicon sequencing, another method for targeted enrichment, will be discussed by my colleague in a webinar next week. So let's deep dive into the workflow. After the library preparation, library molecules are enriched using hybridization capture workflow. Firstly, universal blockers are used to prevent annealing of adapter sequences to each other and to prevent enrichment of targets. Then there is an incubation step with hybridization panel, which consists of biotinylated probes, known also as baits, and they are designed to match sequences of the selected regions of the genome. Target sequences are then bound using streptavidin beads and purified before sequencing. I will start describing our offer for custom panels, and it works in the following way. There is an online tool on our website with an easy interface where user can submit their list of targets in several formats, for example, as a gene list or genomic coordinates, and then request a free of charge design. User can then review the design and order synthesis of panel in one of the 4 formats, which I will discuss in a minute. A dedicated team design team is available for applications in which design might not be possible or optimal with the online design tool, and the assistance from the IDT technical team is also available throughout the whole process of a panel design. Once the panel is synthesized, we then offer NGS functional tests to ensure the performance of the panel, or when the delivery time is crucial, we can also provide a panel format with accelerated turnaround time. Once the user obtains a panel, but still would like to modify it, we can always expand existing panels by adding supplemental probes. When the design is approved by the customer, these are the 4 formats in which we can provide custom panels. Our standard offering, xGen Custom Hype Panel, xGen Custom Hype Panel Axle, formerly known as Discovery Pools, xGen Custom Hype Panel Production, and Production, but without functional testing. All of these offerings include non-binding design service as well as quality synthesis. Our probes, 120 bases long, biotinylated DNA oligos, are synthesized individually at scale before pulling them together, and there is no error-prone amplification processes. And all the panels, they are available in 16 or 96 reaction scale. The differences between these options are following. xGen Custom Hype Panel includes NGS functional test. That means that upon synthesis of the panel, our scientists will use the panel for hybridization capture enrichment and sequence enrich libraries. Results will be then summarized in a report and provided to the customer. If some probes are identified as not performing optimally, we can then rebalance the panel formulation. This offering would typically have 3 weeks of turnaround time. When faster delivery of the panel is required, the xGen Custom Hype Panel Axle would be a more suitable option, as it does not include the functional test nor any additional quality control. When quality documentation is important for the user, we would then propose xGen Custom Hype Panel Production. This format includes functional tests as well as mass spectrometry analysis performed on each individual probe, assuring there are no rarely happening dropouts in the synthesis. Lastly, for recurring orders of production type, we also offer xGen Custom Hype Panel Production with no functional test, only the mass spectrometry quality control. So to summarize, we provide products to span the value spectrum, starting from basic, which is xGen Custom Hype Panel Axle. It has the fastest turnaround time, and it's the best option when speed and affordability is the most important. Our main offering, xGen Custom Hype Panel, has the best trade-off between speed and value, and is the best option when extra evenness and boosted coverage is the most important value for the user. And the premium option would be the xGen Custom Hype Panel production type. It includes additional documentation, so mentioned mass spectrometry control, and this is the gold standard for industry users. So here are some examples of performance of our custom hype panels. The data in A, they show consistent mean target coverage for 2 synthesis lots of certain panel. For the same panel, all 3 lots had very consistent and reproducible on-target rates, as depicted in panel B at the top graph, and coverage at the bottom of the panel B. One of the reasons why our panels are so well performing is the usage of the newly developed off-target QC method that expands target region during the design process. So in this example, a panel of hybridization probes created to capture fragments of human chromosome 21 were screened for off-target hybridization using 2 different QC methods. The repeat annotation RA method was compared to the xGen off-target QC method, which was recently developed by IDT, and it's used in our online tool. And we have compared those methods in their ability to refine a custom hybridization panel that maximize the target region without increasing the amount of off-target hits. So as you can see in the Venn diagram, majority of the example target space passed both off-target QC methods. A little bit less than 36% of the target space passed our newly developed QC method, but only less than 1% of the example space passed only the RA-based QC method. What does it mean practically is that the xGen off-target QC method adds design space with minimal impact on the on-target rate compared to the repeat annotation-based method. One other important aspect of hybridization capture workflow is the increased multiplexing ability. So multiplexing here means using more than 1 NGS library sample for 1 hybridization capture. In fact, here we are comparing 1, 8, and 20 plex with a custom hype axle panel. And as you can see, we observe no decrease of the coverage between the samples. Our recommendation is to use 12 plex for the most optimal results, which means processing 192 NGS library samples with just 16 reaction scale of the panel and hybridization capture reagents. And there is a lot of flexibility with the capture panels as well. To customize a panel, probes can be added to it. In panel A, we are correlating the coverage of a custom panel with and without the spiking of custom probes. And it demonstrates that addition of probes does not impact coverage of the main panel. And in B, I'm showing you some integrated genome viewer, IGV screenshots representing coverage for PIK3CA, which is gene targeted in main custom panel. And then in KRAS, and then PIKs for KRAS, which is additional content from spiking custom probes. And probe regions are indicated in light blue. So as you can see, addition of custom probes does not affect the performance of the main panel. Moreover, xGen custom hype panels, so our standard main option, they are functionally tested prior to delivery. Here we are showing some sequencing metrics obtained in our functional test, and then the validation was repeated by the customer. So as you can see, there is higher reproducibility between the results. And we are offering the functional test, so customer can actually skip the validation process. I've been talking about custom panels so far, but we are also offering off-the-shelf predesigned panels. The first one I would like to focus on is the xGen exome hype panel version 2 for the whole exome sequencing. The panel was validated and used in combination with several of our library preparation methods. And one of them that is compatible with the exome panel is our xGen cfDNA and FFPDNA library prep kit, which I was explaining also on my last webinar. This kit features a proprietary single-stranded ligation strategy and mutant ligase that maximizes conversion and virtually eliminates adapter dimer and chimera formation. Since dimer formation is negligible, a fixed concentration of adapter can be used and the aggressive size selection is not required for post-ligation cleanup. So we have used this kit for some benchmarking studies. Here we have benchmarked our exome V2 panel against exome panels from competitors. The exome panel V2 resulted in the highest on-target rate as compared to exome panels from suppliers X, R, and A. Please notice that IDT exome panel was used in plexing 12 NGS library samples together, and the plexing does not affect much the on-target rate as compared to single plex experiment. The on-target percent is still significantly higher to 8-plex workflows recommended at that time by different vendors. Further on, the exome panel showed the highest percentage of exons covered greater than 20x, 30x, and 40x read depth when compared against the 3 vendors tested. Moreover, IDT exome panel had the most uniform sequencing coverage, superior to providers R and A and comparable to vendor X. Comprehensive coverage is necessary for variant calling, and without adequate coverage, variant calls cannot be made with covariance. So it is important that the low coverage regions are covered, as if they are not, they will need subsequent sequencing, which of course wastes time and resources. In the next point, we tested lot-to-lot consistency. Example coverage of IDT exome panel from 2 different lots was plotted and compared to the competitor X, and it was outperformed by our panel. It was no surprise to us, as our probes are synthesized individually at scale, and we can achieve high lot-to-lot consistency, which allows our users to limit expensive revalidation. Lastly, we have calculated the number of samples loaded into a flow cell. In this example, it was NovaSeq S2 flow cell. As you can see with the IDT exome panel, which allows for 12-plex, you can increase the number of samples sequenced at the same time and therefore reduce sequencing costs. Additionally, our workflow allows for flexibility and customization of the exome panel, and this is also true for any other panel that we provide, either pre-designed or custom. The flexibility comes from the fact that we synthesize probes individually, and that allows users to order specific probes to supplement any existing panel. These supplement probes can be used to rescue regions that are either poorly captured by existing panels, and it's done by enhancing their performance, or they can be used to extend the target region of the existing panel. We have also some predesigned panels that were designed with a purpose of using them as a spike-in panels, which I will discuss in more detail in a couple of slides. And one example of such spike-in panel used together with the exome panel is presented here on this slide, where we used CNV copy number variation backbone panel as a spike-in to xGen exome panel. Additional information from intergenic SNPs targeted by the CNV panel improves the resolution of a deletion in PTEN gene, shown in this example IGV snapshot. I will now finish talking about xGen exome Hype panel v2 by just providing a few facts about the design. The panel was designed against the RefSeq 109 annotation from January 2019, and it is native to reference genome HG38. The exome panel version 2 targets coding regions of around 50,000 known RefSeq transcripts, which corresponds to 19,433 genes with a footprint of 34 megabases. The panel consists of more than 400,000 probes manufactured under strict ISO 13485 standards, and it is available in 4, 16, and 96 reaction scale. I will now move on and briefly describe other predesigned hybridization capture panels available at IDT. Each of these stock panels can also be customized if additional regions need to be added. And some panels, such as the human ID, human mitochondrial DNA, and CNV backbone panel, are also routinely used as a spike-ins to the exome or other panels, but can be also used as a standalone panel if needed. So firstly, the AML panel targets more than 260 recurrently mutated genes associated with acute myeloid leukemia. Some of these target genes are listed in the table, and full target list is always available on our website. When validating the panel, we have consistently obtained high coverage uniformity as presented in the data here. Greater than 0.2 mean coverage is observed for more than 98% of the targets. We can also achieve deep coverage of targeted regions with the AML panel. Here in all samples, more than 30x coverage was achieved for more than 98% of the targets, and this was only slightly lower percentage compared to more than 1x coverage. And lastly, we obtain consistent results with various lots to lots of the xGen AML cancer panel. Next panel that I will very briefly discuss is the xGen pan-cancer panel, targeting 127 genes found to be significantly mutated in 12 cancer types. Genes targeted by pan-cancer panel are important in several cellular processes listed on the slide. And we have also, of course, performed validation of this panel. So example of performance is shown in this slide. The panel provides almost complete coverage across all targets, with 99% of targets covered at 30x coverage. A pan-cancer panel provides also highly uniform data, as shown in this figure, that presents the percentage of targets that are covered and indicated mean coverage. Finally, pan-cancer panel as well shows high correlation between different lots, represented here as a correlation of depth of coverage obtained with 2 different lots. Next panel is xGen Inherited Disease Hype panel. It enables sequencing of genomic regions containing genes and SNPs associated with inherited diseases. Traditionally, whole exome sequencing has been the standard method for detecting a wide variety of inherited diseases. However, sometimes we have users that they would prefer focus sequencing of specific genomic regions of interest to have deeper coverage for easier, more efficient, and more accurate coverage of disease-causing mutations. The inherited diseases panel targets all disease-causing mutations as defined by the human gene mutation database, combining the use of whole exome sequencing with the low per sample cost of targeted panels. We have also performed a bunch of validation experiments with this panel, but for the sake of time and not to bore the audience too much, I will not be showing them today. I will now move on to 3 panels that are usually used as spike-ins to the exome or other panels, but can also be used as a standalone panels. The first one is xGen Human ID panel, capturing 76 distinct, highly polymorphic sites. The Human ID panel enriches a definitive set of SNPs selected to identify a unique individual from a population of over 5 million biological samples. The panel has been tested with DNA from FFP tissues, cell-free DNA, and other sample types. The best applications of this panel includes assessment of tumor versus normal paired samples, tracking and management of individual samples during longitudinal studies, and identification of mislabeled or misidentified samples. Here I am showing an example of the data proving that the spike-in of the Human ID panel does not impact on-target performance of the recipient panel. In this case, the recipient panel was a custom panel containing 509 probes. And here it is represented by showing mean on target percentage for the custom panel alone, and with Human ID panel spiked in gray. Next spike-in panel is the xGen Human Mitochondrial DNA panel. It is designed to simplify the investigation of the human mitochondrial genes, and it provides coverage of the entire 16 kb human mitochondrial genome. We have also spiked in this panel to the custom panel mentioned 2 slides ago, composed of 509 probes. And the same as for the Human ID panel, we do not observe an impact on the on-target rate of the recipient panel. If something, we even see slight increase in the mean read on target percent. Lastly, our third spike-in panel that can be and is also used as a standalone panel is the copy number variation backbone hype panel. It spans the entire human genome with probe space approximately every 0.34 megabases. And it allows to generate reference coverage data that can be used to detect copy number variations. And this is the last predesigned panel I wanted to talk about. I will now move on and briefly explain other reagents used for the hybridization capture workflow of IDT. After the library preparation and before the enrichment with hybridization capture panel, universal blockers are used to prevent annealing of adapter sequences to each other and to prevent enrichment of off-target by reducing non-specific adapter interaction. IDT blockers are universal and not sequence specific, and we provide them in 3 standard versions. There is TS-mix for libraries that contain true-seq adapter sequences with 6 or 8 bases long index. 10 BPTS mix for true-seq adapter sequences with 10 bases long index. And finally, NXT mix for libraries that contain Nextera adapter sequences for either both 8 and 10 base long indexing. And all of the options are available in 16 and 96 reaction scale. When using IDT library preparation kits, we provide guidelines for compatible blocker. If libraries are prepared with other vendors' kit, it is important to pair adapter with right blocker, and we can also support in this decision. So why it is important to use blockers? We highly recommend it to include them in the hybridization capture reaction, as they significantly increase on-target rates and reduce off-target rate at the same time. For example, I'm presenting here data showing the mean on-target percentage for 2 predesigned panels, AML and Human ID Research Panel, both used as a standalone panel and used in hybridization capture workflow with addition of blockers or without universal blockers, so here labeled as no blocker. I think you can appreciate the positive impact on the on-target rate when the blocker is used. And of course, it goes without saying that preventing adapter cross-hybridization reduces cost of sequencing by increasing the efficiency of the sequencing run. Apart from mentioned off-the-shelf universal blockers mixes, we also provide custom blocking oligos compatible with other platforms, including Ion Torrent and for other specialized applications. Next component of the hybridization capture workflow is the xGen hybridization and wash kit. This kit includes all necessary buffers and reagents to perform the capture. It includes streptavidin beads as well as human code DNA to block non-specific binding for repetitive DNA sequences in the human genome. As usual, we have thoroughly optimized the buffers composition and internally tested the reproducibility between various buffer batches. As you can see here, we can assure reproducible results between different experiments using different lots of our kit, even when different users perform the experiment. So here, 3 different users has performed the experiments. Last product to be included in the hybridization capture workflow next to panels, blockers, and buffers is the xGen library amplification primer mix. And the primer mix simply contains forward and reverse primers that target the P5 and P7 regions of Illumina sequencing adapters. They are premixed and formulated in ready-to-use concentration and are used for post-capture PCR step in the protocol. As a final comment, I would like to mention that the PCR polymerase has been so far required to be purchased separately, but as of September this year, so this is a very fresh update, the xGen Hi-Fi polymerase is now part of the kit. And hopefully that simplifies the ordering process, as there is no need to source components from multiple vendors. And this was my last slide. So thank you for your attention, and I will be happy to take any questions now if there are.
Okay, I don't see any questions in the Q&A. Also no questions in the chat box. Maybe we can just wait a minute or two. Thank you very much, Julia. Okay. Okay, so we have one question. The question is, please comment on automation options. So is the workflow automation compatible?
Yes, workflow is automation compatible, and we have in-house several scripts that were developed for various machines. So maybe and there is a various testing done on these scripts. Either it's with reagents or just water. So we have those scripts. Some of them are more developed, some less developed. We can also provide support for automation if we do not have any script available. But the list is long and I don't have it by heart. So I think the best option would be to actually provide the information, what kind of automation platform from what type of provider user has, and then we can discuss what can be done. Because it could be that we either have a script ready or script is under development, or maybe we don't have an option, but we have some other users who develop this script, and we can, usually they are very open also to share their experience. So the list is long, as there are many automation providers. I think the best is to provide information what kind of platform the user has.
Thanks, Julia.
So feel free to contact also Whitehead Scientific or IDT directly for this information.
Yes, that is definitely something that we are able to assist with, that we can help with. We have application specialists as well who are in the various regions, you can assist with this as well. So if you have a specific automation system that you would like us to help you with this particular workflow, or alternatively, if you need an automation system, that is also something you can talk to Whitehead Scientific about. Okay. Okay, I don't see any other questions. Right. So if you still have any questions that have not been addressed today, or if you haven't been able to share them with us, please feel free to send them to a Whitehead Scientific representative, and we will definitely be able to assist you. We can put you in touch with our application specialist, or we can reach out to Julia as well. Okay.
That's good.
Thanks very much, Julia, for presenting today, and thank you to all of our attendees. And we hope to see you on the next webinar soon, next week. The last one in our 3-part series. Thank you very much.
Thank you very much.
Bye.
Bye-bye.